gene structure, there are a few more words to know here. So I did mention before introns and non-coding sections of a gene, or so-called interrupting regions. Exons are sections of a gene that form mRNA and code for proteins, or they are the expressed regions. And you've got the promoter region, which is a section of gene which RNA polymerase can bind to. Operator regions are found in prokaryotic operons, which we'll look at in a few slides, but these are not in eukaryotic genes. So how come different cells produce different proteins even though every cell contains the same genome? Well, pretty much cells can switch genes on and off, and this is called upregulating or downregulating. And so they can up or downregulate particular proteins based on the specific needs of a cell. So you can see here, in this muscle cell, we definitely need muscle protein, and so we might actually express gene A, which might be really important in the muscle cell. We don't need gene B perhaps, and so we can downregulate it, and we can upregulate gene C just a little bit. And this may be exactly what we need for the muscle cell, but you can see that the needs differentiate in the skin cell, and that we actually don't need so much expression of gene A in the skin cell. We do need quite a bit of B, so we'll upregulate B, downregulate A, and probably downregulate C as well. And then for the third one, you can see that you definitely downregulate gene B for the nerve cell. So really just kind of switching these genes on and off as needed in order to facilitate and kind of ensure that you're looking at the particular needs of a cell. So how are genes turned on and off? So we've got lots of different transcription factors, and what these are binding proteins, or DNA binding proteins, which bind to specific sequences of DNA at the promoter region and other regions as well, and they regulate gene expression, and they can either increase or decrease transcription of a gene. So things called repressors, these are transcription factors, and they prevent transcription by binding to the promoter region or the operator region of DNA, thereby preventing RNA polymerase from binding and transcribing the DNA. So you can see you've got activators binding to enhancer regions, and this leads to high levels of transcription. You may have repressors binding to operator regions, and this may suppress transcription and actually prevent transcription from occurring, thus downregulating a particular gene. So how are genes turned on or off? You've got a few other genes. You've got regulatory genes. These genes encode for the production of proteins, such as transcription factors, that regulate gene expression. And you've also got structural genes, and so these are genes that produce a protein or product that forms part of the structure or the functioning of an organism. Essentially every gene except regulatory genes. You've got signaling molecules. So signaling molecules such as hormones can bind to receptors on or within a cell, and they can influence gene expression in that particular cell. So a lot of signaling molecules such as hormones are actually proteins, and so they can activate transcription factors that assist in RNA polymerase binding. They can inactivate transcription factors that prevent RNA polymerase binding, can inhibit or switch off particular genes, and also bind DNA directly, and this can alter gene expression also. I'm like dehydrated. Get more water. So gene expression is influenced by cell type, stage of development, and the external and internal environments of a cell.